See, there is a genetical phenomenon called as epistasis. In this presentation, I will explain what is epistasis. Particularly, I will explain about recessive epistasis because epistasis could be of so many types. There could be dominant epistasis or double dominant epistasis or recessive epistasis. Sometimes there may be a situation where you may observe double recessive epistasis. So it is of so many different types, and in all such cases the Mendelian dihybrid ratio, that is 9331, gets modified to a different kind of ratio. So epistasis is a particular phenomenon in which one gene masks or prevents the expression of another gene. Okay, so you should understand in this way that there are two gene loci. They are situated on separate chromosomes, but one gene, and both are responsible for the expression of a single trait. In such situation, when one gene prevents or interferes the expression of another gene, then that phenomenon is referred as epistasis. And because of this interference, the Mendelian classical dihybrid ratio 9331 gets modified. So, in case of recessive epistasis, the phenotypic expression of one gene is suppressed due to the presence of homozygous recessive allele of another gene. This is important point to understand, that when one gene is in homozygous recessive form, in such situation it is masking or it is suppressing the expression of another gene. And because of this, the classical ratio 9331 is modified to 934. So when we find such ratio 934, then it is because of recessive epistasis. And this phenomenon has been recorded long back in case of mice coat color. So we can understand by taking this example, in which you find that in case of mice, agouti color is the wild type of color. Actually, it is a grayish color of the hair. Mice which have grayish hair color. Actually, the appearance of gray color in the hair is because of some banding pattern. So such mice are called as agouti. And those mice which do not possess color at all in the hair, they are actually albino. So when we consider the agouti color, it is due to the presence of two genes present on separate areas on the chromosomes. And suppose there is a gene capital A. This capital A causes agouti color. Okay, capital A is responsible for the appearance of agouti color, and it is dominant over other color like black color. And when this capital A allele is in recessive homozygous form, then it causes black color. So capital A, that is dominant over small a, capital A is causing agouti color, and small a is causing black color. This point one has to understand. And then capital B is means B is another gene locus, and in that gene locus, if there is recessive homozygous condition, that is small b, if this situation is there, then it is epistatic to capital A or small a. Means this gene, when homozygous recessive small b, when this genetic makeup is there on a specific gene locus, then the individual will neither be agouti nor be black, because this small b is epistatic to capital A or small a. So we can have this idea by taking this particular case in which agouti, whose genotype is capital A and capital B, means on both gene loci the dominant alleles are present, and this mouse is being crossed with another mouse who is albino. Albino means that individual is lacking color pattern at all, that looks white in color. And genotypically, this fellow is small a, small a, small b, small b. Means on both gene loci, recessive alleles are there in homozygous condition. So the parental cross is made like this, and then in F1 generation, you will have all agouti, whose genotype will be capital A, small a, and capital B, small b. Okay, so you have agouti males and females. They are genotypically heterozygous on both gene loci, capital A, small a, capital B, small b. Now let us cross these agouti and see what would be the situation in next generation. So here in this uh, figure, we observe that two agouti individuals who are heterozygous on both gene loci, they are being crossed. You know that since these two gene loci are present on separate chromosomes, so there would be independent assortment of gametes, and you will have four different type of gametes in male individual. Exactly the same type of situation will be there in female individuals. Females will also have four different types of gametes. So such gametes will be capital A, capital B, then capital A, small b, and then small a, capital B, and small a, small b. These four different types of male gametes are formed. In the same way, four different types of female gametes will be formed, and there would be random fertilization of these gametes. So you will have 16 different kinds of combination. But see, out of these 16 different kinds of combination, you will have 9 types who will have agouti coat color. So see, all those in which capital A and capital B alleles are present. In such cases, in all such cases, like these are heterozygotes, these four are heterozygotes where capital A and capital B alleles are present on both the gene loci. So these 9 types who will have capital A and capital B, they will be agouti. But those who have capital AA and small bb genotype, such individuals are actually albino. Why this is happening? Although capital A is there, this is a dominant allele, and it should cause a gouty color. But this fellow, this individual, is actually albino because the small b alleles are epistatic to capital A. Okay, so such individuals are albino in their phenotype because small b combination that suppresses the expression of this agouti color. Exactly the same way this fellow, I am showing with this indication that this fellow will also be albino because here small b alleles are there. And this fellow will also be albino, and this fourth one will also be albino. We have already used this type in the parental cross, so they were albino. So 1, 2, 3, 4, these four types will be albino, and these three types will be black in their color because they are small a, and such small a genotype that causes black color, and this capital B is not epistatic, so its presence is actually required for the expression of black or agouti. So you can say that this fellow is black in phenotype, this one is also black in phenotype, this mouse will also be black in phenotype. So instead of getting 9:3:3:1 ratio, we shall be getting 9 agouti and 4 albino and 3 black. So in this table you can see the F2 ratio 9:16 are those individuals who have capital A and capital B alleles are on both gene loci, they are agouti types. So finally they will be 9:16. Then those individuals who have capital A and small bb, such individuals are 3 out of 16, they will be albino. And those who are having genotype small aa and capital B on a different locus, such individuals are also 3 by 16, they will be black in their phenotype, and small aa, small bb types, they are albino. So in total 9 by 16 will be agouti, and these albino, that is 3 by 16 and 1 by 16, means 4 by 16 will be albino in their phenotype, and 3 by 16 will be black in their phenotype. So we observe here in this particular case that recessive alleles in homozygous condition and at one gene locus may be epistatic to a gene which is situated on a different chromosome. And we can cite the example of recessive epistasis by taking other cases in case of human as well as in case of Drosophila. There are several examples which are very appropriately cited to explain recessive epistasis.